Culturing microorganisms is another thing you need to know for your GCSE Biology exams. And make sure you let me know in the comments of any other topics you want me to go through with you. Now, culturing microorganisms involves growing bacteria or microbes in controlled conditions to study the effects of things like antibiotics. So let's go through how we'd firstly grow the bacteria medium. And I really want you to remember that we need to work aseptically here, which means we want to avoid any contamination of our medium. So as I go through this process, I'm going to talk you through the aseptic techniques as well, and I want you to remember these for your exams. So as we begin this experiment, the first thing we want to make sure is we have a Bunsen burner which is lit with an orange flame. And the reason this is because the Bunsen burner will create a convection current, which means the air around it, our workspace, that air will rise because warmer air rises, and it will take away any microorganisms in the air in our workspace up and away from what we're about to do. That's an aseptic technique. The second thing we would do is we would pour hot agar jelly into a sterilized Petri dish. Now notice the Petri dish is sterilized, aseptic, and the agar is hot, so any bacteria will have died. Aseptic technique as well. Then we have an inoculating loop, which looks like this. We pass this inoculating loop through a hot blue Bunsen burner flame to make sure any microorganisms on it are killed. Aseptic technique here. Now this next bit's going to require a little bit of imagination. Imagine that this is my Petri dish. Open, close. This is my inoculating loop made from micromire, and I've already dipped it into my Bunsen burner flame, so it's already decontaminated. I'm going to put this into my bacteria. I'm going to ever so slightly open up my agar plate, and I'm going to inoculate the agar jelly with this bacteria. As soon as I'm done with that, I'm going to close this Petri dish and I'm going to tape it shut. That's another aseptic technique that you need to remember, because by only opening up the agar plate very little, we're avoiding any air going into it, or as little air as possible getting into it, and therefore any bacteria, microorganisms in the air from getting in as well. Once we have done this, we can then incubate this Petri dish at no higher than 25 degrees Celsius, because at temperatures higher than this we can have dangerous bacteria growing. I'll do another video showing you guys how we can calculate the effectiveness of antibiotics and calculating their zones of inhibition. So make sure you guys like this video, send it to a friend, and check out our GCSE predicted papers on christuition.co.uk. See you guys on the next one.